All right, so we're ready to move on to part 2, and the goal of part 2 is to figure out what each of these spots are from part 1. And so on the data sheet for part 2 on the lab handout, what we're going to do is use the ideal solvent ratio to run another TLC plate. Now you should be able to figure out what the ideal solvent ratio is, because it's the one in which all the spots that were on the TLC plate spaced out from one another as far as possible. And so you'll be required to fill in and determine what the solvent ratio, what the best solvent ratio is from part 1. And then what we're going to do is fill in the names of the compounds in the unknown and figure out its identity. So what we're going to do is take a much larger TLC plate. Again, we draw our baseline across the bottom of the TLC plate. And this time we're actually going to run 5 samples at once on the same TLC plate. And so what we do is we create these different lanes. We draw a tick mark spaced apart from another tick mark, and we label it with some sort of letter or number. And so the first tick mark that I drew, that's where we're actually going to spot unknown number 3. The second one, which I drew as letter A, that represents acetaminophen. And so what we're going to do is run a pure sample of acetaminophen. Then the next tick mark, I drew AA, representing acetyl salicylic acid. We're going to run a known sample of acetyl salicylic acid, see how far that travels up the TLC plate. Then we're going to have a lane for caffeine and a lane for diphenhydramine hydrochloride, the hydrochloride salt of diphenhydramine. So because we have a much larger TLC plate, we need a much larger solvent chamber to be able to place the TLC plate in. So I already filled our solvent chamber, our beaker, with our ideal solvent ratio, which you will have to determine. And I'm going to place my TLC plate into that solvent chamber, into the beaker, and place a watch glass on top. And then again, just like in part 1, we're going to see the solvent carry the compounds up the TLC plate. And once the solvent line gets close to the top of the TLC plate, we'll remove the watch glass and pull the TLC plate out of the beaker. All right, so this is what our TLC plate looks like after I illuminated the spots with UV light and circled them with pencil. And so all I've done was recopy that onto our data sheet here. And so what you want to do is look at the unknown number 3 lane, and we see that we have 3 compounds that separated in that lane. They all traveled different distances up the TLC plate. And what you want to do is try to match up which spot is which with the known standards that we did run on the TLC plate. And so next to the unknown number 3 lane, we ran acetaminophen. Then the next lane, we ran acetyl salicylic acid. Then we ran caffeine, and finally diphenhydramine hydrochloride. And so what you want to do is name the 3 compounds that were contained in this unknown, and then determine the actual commercial drug. And so the 3 options we have are Tylenol PM, which contains acetaminophen and diphenhydramine hydrochloride. We have Excedrin Extra Strength, which contains acetaminophen, aspirin, and caffeine. And finally, we have Excedrin PM, which contains acetaminophen, aspirin, and diphenhydramine citrate, which would run the same amount of the TLC plate as diphenhydramine hydrochloride. So your task is to determine the identity of the unknown. It would be one of the 3 drugs that I just showed you.